Up next, we have Dr. Sarah Teichmann, who was a distinguished leader in cell atlas technologies and cellular genetics. Dr. Teichmann established her research group at the MRC Laboratory of Molecular Biology in 2001, and then moved to the Wellcome Genome Campus in 2013, becoming the first faculty member appointed across both the EMBO European Bioinformatics Institute and the Wellcome Sanger Institute. In April 2024, Dr. Teichmann joined the University of Cambridge as chair in stem cell medicine, while also working with GlaxoSmithKline and her startup, EnsoCell Therapeutics. She co-founded the International Human Cell Atlas Consortium, which now spans over 3,000 members, aiming to map all human cell types. Dr. Teichmann has received numerous prestigious awards, including the EMBO Gold Medal and the FEBS EMBO Women in Science Award, and is a fellow of both the Academy of Medical Sciences and the Royal Society. Please join me in welcoming Dr. Sarah Teichmann.
Thank you for that kind introduction, and thank you for having me. It's a tremendous honor and a pleasure to speak today in this symposium for the Next Generation Sequencing Canada Gairdner Award. And I want to just give a little anecdote that makes it particularly special for me to be here, which is that almost exactly 30 years ago, Shankar taught me organic chemistry when I was an undergraduate, second-year undergraduate. So I don't think at the time either of us thought we'd be meeting here again, but great pleasure and delighted to be here. And of course, Shankar went on to discover next-generation sequencing with David Kleinerman and Dr. Meyer. And I went off and had an exciting journey through theoretical and computational biology. And that's what I'm going to tell you about today, about molecules and cells. So mapping cells is an idea that has a long history and has sort of probably popped up in different places in the world again and again. One of the lectures that kind of is on the internet and is famous is the Nobel lecture by Sidney Brenner, where he says, we need a program of making maps of cells and maps of how cells talk to each other. The Cell Map project, for which we don't need a model organism, because of course we can study ourselves, the human body, will be one of the things to occupy us for the next few decades. And what Sidney didn't know was that there was going to be a resolution revolution in genomics. So I don't mean next generation sequencing, but what I mean is actually the ability of processing cells and the library preparation step upstream that allows us to sequence the nucleic acid content of individual cells. And you already heard in the amazing previous talk on epigenetics how important it is to understand cell states. And you can see that with this technology, basically it's possible, from the earliest days, which were sort of pioneered by Azim Surani and others, studying a handful of cells, this technology underwent a scaling up where it's now possible to study hundreds of thousands or millions of cells in a single experiment. And so what that means is that you can take a tissue sample, like a human heart sample, and then study the individual cells or nuclei in terms of the transcriptomic content of the cells. In other words, the RNA that's expressed, the genes that are switched on in that single cell. So we've heard a lot about the DNA sequence of individuals in health and disease. We've heard a lot about the epigenome. And of course, all of that is encoding the repertoire of genes that are active in single cells, which are very different in a cardiomyocyte versus an immune cell versus a fibroblast versus a neural cell, et cetera. And so sequencing at the single cell or single nuclear level in suspension gives us that huge amount of information about which genes are active in each cell, which we can then analyze with computational methods, deep learning, AI/ML, and so on that we have available nowadays. And hand in hand with that resolution revolution, what we've had is technology development in spatial genomics. And spatial genomics, it sort of comes through the sequencing side and the imaging side, and sequencing and microscopy are sort of coming together again in this spatial biology field, which is kind of also raced forwards in the last decade or so, where we can then map in a tissue section, so taking a salami slice of a human tissue sample, the gene expression landscape of the cells in that section. And then you can imagine combining that computationally with the deep knowledge of the cells in that sample, with consecutive sections, we can build up a picture of the tissue niche in that sample, in that tissue, and understand how the cells are oriented relative to each other. And this spatial biology, is really becoming kind of, widespread, prevalent, and popular. And earlier this year, in June, we had a small symposium in Stockholm called Spatial Biology in the Genomics Era. And that's really the era that we live in. The era that we live in now is single-cell genomics and spatial genomics. And Aviv and I, Aviv Regev, both gave talks in the last section. Of course, she's the co-founder with me of the Human Cell Atlas International project that Rob mentioned at the beginning of the talk. And so why is this mapping important? Why is it important to know in exquisite detail and at full molecular depth the fine cell states in a tissue sample and how they're located relative to each other? It's important because it's not just a kind of enumeration and mapping exercise, but that information also tells us about the function of the individual cells and the cell states, because it tells us how the cells are talking to each other. And the molecular dialogue, basically, between the cells, it tells us about the cell function and therefore then also the function of the tissue within our organs. And to illustrate that, I'm going to tell you 3 short stories about 3 different tissue niches: the placenta, the heart, and the thymus, if I get through them. So why is the placenta so fascinating? It's a transient organ that forms at the beginning of pregnancy. And of course, it's necessary for delivery of oxygen and nutrients to the embryo. What fascinated me about it is that conundrum that Kathleen presented earlier, which is that our T cells and our immune system is trained to distinguish self from non-self. And of course, in the placenta, just like in a tumor, the maternal immune system is presented with the paternal antigens. So they should be, in theory, the maternal immune system should be rejecting the paternal antigens, but instead what happens is that there's a remodeling of the tissue so that you have these larger arteries that are formed to deliver the oxygen nutrients through the blood to the placenta, and essentially a harmonious coexistence of the fetal cells that are the invasive trophoblasts that are entering the uterus and the maternal cells on the uterine side of the placenta. And Janet knows this well because she's one of the discoverers of the trophoblast stem cells. So the conundrum is really, how does the maternal immune system respond, and what are the interactions that allow this coexistence, this peaceful coexistence? And to address that question, Rosa Venter-Tormer, who was a postdoc in the group, set out on this brave quest to single cell sequence the cells both on the maternal and on the fetal side of the first trimester placenta that we got from the Human Developmental Biology Resource in Newcastle, with the support of Mus Hanifeh's lab, who is right next to the ward there, and also Ashley Moffat's group. And this allowed us for the first time to map out the fetal genotypes because Mus had also gotten research ethics to get the maternal blood, so we're able to use the sequencing data to say what is maternal, what is fetal, and distinguish macrophage states, for instance, that were fetal and macrophage states that were maternal. So distinguishing which cells in this intermingling come from the mother and which come from the fetus. And this sort of dialogue between the cells that's absolutely crucial to get right at this interface in order for the remodeling of the uterus to allow the blood supply to form correctly is an interaction that we deciphered by mapping the molecules on the surface of the cells and then statistically inferring what are the molecular interactions, what's the molecular communication between the fetal and the maternal cells. And you can see here there are 3 natural killer cell states that we described in molecular detail for the first time in this sort of continuum of activation of natural killer cells, and these are obviously cells that normally kill non-self. So how are they kept in check? They're kept in check by the particular receptor-ligand interactions that they see on the fetal side with the extravillous trophoblast, and from the macrophages and stromal cells on the maternal side that are also kind of controlling these cells and keeping them calm, as it were. And where does the, what was the thinking here behind these receptor-ligand interactions, basically in the cells? So combining the cells with their molecular cell surfaceome and their interactions, well, that's from basically the 15 years that I spent at the MRC Laboratory of Molecular Biology in the Structural Studies Division, where we were studying protein biophysics. And you can see a few years ago, we had a Nobel Symposium with these gentlemen here, again in Stockholm in 2022, Demis Hassabis, who was, of course, awarded the Nobel Prize for the AlphaFold. And what we had worked on was modeling protein complexes as graphs and understanding the assembly pathways of proteins in 3 dimensions and how they find each other inside cells. And also, of course, then what we were asking here was how do they interact with each other on the extracellular surface in a stoichiometrically accurate way with all the subunits there at the right concentration. So we're not thinking of protein-protein interactions as binary interactions. We're thinking of them basically in terms of all the subunits that are needed that we know from protein structure and protein biophysics. And so it's really bringing together this molecular kind of thinking about proteins and the cellular interactions that led us to a deeper understanding of the placenta, of the cell types and developmental trajectories of the cell-cell interactions and how that immune tolerance is set up. So that's really a story about our development, the human development during pregnancy, which is, by the way, different from the early kind of developmental stages in mouse and the molecules in the mouse. So there's a lot of evolutionary divergence. So this concept that Sidney Brenner said of not studying a model organism but studying ourselves is really quite relevant when you want to really get to the molecular details in the tissues. So humans are very different, both in terms of the immune system in particular, which is obviously evolving very fast because of host pathogen arms race, but also speciation kind of in these early developmental tissues. So next, I'm going to go on to a different organ that's incredibly important to life, which is the heart, obviously, one of the crucial organs, and one of the most complex organs in our body besides the brain. And I want to talk about, again, tissue niches, but also cell-cell interactions, both in a paracrine sense, but also in an endocrine sense, which means signaling. Endocrine is the hormone system, and signaling between distant regions of our body and coordinating our physiology. So we started studying the human heart with collaborators in Berlin and Boston many years ago, and initially using this suspension cell, single-cell genomics technologies to define the cell types in the free walls of the 4 chambers of the heart. So we've got 2 atria, 2 ventricles, and defining the kind of muscular tissues in terms of both the cardiomyocytes, but also the immune cells and fibroblasts and so on. And the complexity that we discovered was quite shocking. So instead of having, let's say, 1 cardiomyocyte subtype in the ventricles, we found 6 or more. And then more recently, what we moved on to was wanting to understand not just the muscular tissues in the heart, but also the sort of brain of the heart, which is the cardiac conduction system. And that electrical conduction system basically maintains the coordination between the contraction of the 4 chambers, the muscular tissue in the 4 chambers. And that beating of the heart, that rhythm and that coordination is kicked off by so-called pacemaker cells. So pacemaker cells are very special, very rare cells in our body. They're amongst the only spontaneously firing cells, and they start beating very, very early in embryonic development and are kept beating through a sort of clock of calcium and potassium channels that goes on forever until, of course, we take our last breath and the heart stops. So these are absolutely crucial cells to understand. They're very hard to find because they're so rare, and the surgeon who's doing the dissection of the tissue for us wasn't able to see them, also because the tissue that they sit in, the sinoatrial node, is located in different positions in different people and isn't obvious, isn't always in a perfect stereotypical position. So we worked with a cardiac pathologist, Professor Yen Ho in London, and the team, James Cranley and Kazumasa Kanemaru, who are both clinician scientists, trainees in the group, were absolutely brilliant in catching these very rare cells that you can see here in blue at the top. These are from 2 donor hearts that we were lucky to have donated during the pandemic when the transplant was down in the UK. So James and Kazumasa traveled to different cities in the UK, London, Newcastle, so on, to get these declined donor hearts and then do the dissections and then send the tissue to Yen, the images for looking at and identifying these regions. And you can see this tiny cluster of cells has a different profile in terms of the sodium channel expression that's high in these working cardiomyocytes in the right atrium. And but they have very high expression of this calcium channel, CACNA1D, which has been described from murine cells as typical of pacemaker cells. And here what you can see is the profile of all ion channels and G-protein coupled receptors in cardiomyocytes and neural cells, neurons and glia in the heart. And you can see that these cells have a special profile that's different from the working cardiomyocytes and the neural cells. And that gave us confidence that we had actually caught those very special cells in the human sinoatrial node for the first time. What we did then was combine that with the spatial genomics that I mentioned, and indeed they mapped to the node, to the nodal region there that you can see. And that's at the center of this region where we see the inner nodal region with the pacemaker cells, and then we define with the spatial genomics outer nodal regions that has fibroblasts, adipocytes, and macrophages that are kind of electrically insulating these cells. And what we were also able to decipher was the niche of the pacemakers with special glial cells that we were able to define for the first time that are kind of hugging the pacemakers and sort of nourishing them and connecting to the sympathetic nervous system, which is the parasympathetic nervous system that comes from our brainstem via the cardiac ganglion to kind of keep down the heart rate, and the sympathetic nervous system, which says go faster when we see a bear, and so modulates the heart rate in that way. And so then, because heart rate, kind of as I've indicated, is so important kind of in modulating our physiology, what the 2 clinician scientists who are working on this, James and Kazumasa, kind of wanted to know is which drugs can we predict to be acting on the pacemaker cells. And so I said, go next door to the European Bioinformatics Institute, where there's the Chembl database. This is an open source database of drugs and targets, and map those, develop drug to cell, which they did with Krzysztof Polański and others in the group, and map those drugs to all the cardiac cells, including the pacemaker cells. And what was not surprising was that all the chronotropic drugs have their receptors in the pacemaker cells as expected, but then there are other drugs that we found we predict to be acting on these cells because there are receptors in the cells. And the most surprising was the GLP-1 receptor to us, which is of course the target of GLP-1 receptor agonists, liraglutide, semaglutide, which are the Wegovy and Ozempic and Mounjaro and so on drugs. And indeed, if you look at the clinical real-world evidence, there's a 6 beats per minute increase in heart rate initially when these drugs are given. So there's clinical real-world evidence that these holds, and we also show in vitro in experiments in the dish with fetal cardiomyocytes that you get a change in the beating pattern when you add GLP-1 receptor agonists. And recently there's been a paper from a pig showing the same thing. And so this allowed us to hypothesize that basically the 6 beats per minute change comes through a direct mechanism on the pacemaker cells rather than an indirect mechanism via the brain or the autonomic nervous system. And so this basically gives you an idea that the human cell atlas, which is what we're contributing to here, where we're building the cell atlas of the human tissues, gives us new insights not only into signaling in the tissue niche, paracrine signaling, but also endocrine signaling. These are hormones and how hormones are acting across the body in new and unexpected ways. And so that's also incredibly exciting. So to summarize here, we see the new niche for the cardiac conduction system with the autonomic nervous system. Drug to cell kind of shows us that we can use this data for toxicology, repurposing, and for reconstruction of both paracrine and endocrine signals. So one last story in the last few minutes is the thymus, and this is relevant because, of course, for this prize this year of the Gairdner Awards, there's also the CAR T cells are being awarded, and T cells develop in our thymus. The thymus is a little tiny organ next to the heart, was discovered as an immunological organ in the 1960s. You may or may not have heard of it. It's probably one of the most recently discovered organs in the human body. And the hematopoietic stem cells come to the thymus. The T cells develop and are trained to distinguish self from non-self in the organ. And they then go around and colonize the body. And of course, those cells also have therapeutic potential. So the question with this little tiny organ, which is much smaller than the heart, and it actually involutes with age and becomes even more smaller during puberty, is can we map, can we atlas an entire human organ? And how can we do that? By combining multiple modalities together from single-cell genomics, spatial genomics, and multiplex protein imaging. And the way we figured out that we can do this is to model the organ onto a quasi sort of 2.5-dimensional framework using a linear model from the outside to the inside of the organ. And so the concept here is very simple. We form an axis, and you can think of the lobes of the thymus like an egg that has an egg white on the outside, that's the medulla, and an egg yellow on the that's the cortex, and the egg yellow in the middle, that's the medulla. And what we're modeling is each lobe of the thymus as an egg white, sort of from the outside to the inside, with landmarks and then a nonlinear modeling between each landmark to give us an idea of where each data point is coming from the spatial genomics, the single-cell genomics, and the multiplex imaging that we did with Ron Germain's group. And what this allowed us to do then is to build up a model of the organ in terms of all these different layers. You can think of them like layers of an onion, from capsular to subcapsular, through the cortex, through to cortical medullary junction, and into the medulla, which is the center of the organ. And what this allowed us to discover was new insights into precisely how this cellular development is taking place from the hematopoietic stem cells, or the early thymic progenitor, that actually enters inside the organ kind of unexpectedly. And then we show that it travels basically around up to the outside of the organ, and then the cells re-enter the medulla and are trained. And sort of quantifying that in a precise way revealed new, unexpected positionings of the cells as they go on this journey of maturation through the organ. What it also allows us to do is to define precisely what the cells are seeing in terms of the extrinsic signals as they go through this journey. And they are receiving basically cytokine signals, they're receiving growth factor signals, and we can quantify this in terms of the different layers of the onion and what the epithelial cells, the fibroblasts and so on, are expressing, the dendritic cells at each layer in terms of these factors, and then also show how this differs in fetal stages of thymic development, so during pregnancy, versus pediatric stages of thymic development when we have the mature organ. And that's important because the T cell output is slightly different during organogenesis, when the thymus is forming during pregnancy, and the T cells actually contribute to development of our tissues, versus after birth when we have to fend off infections and other challenges. So the nature of the cells kind of changes. What we were also able to do was to map the identity or the the cell state of the cells as they then travel to peripheral organs, and this is using tissues from the fetus. So just to sort of summarize, we're understanding and mapping the T cells across scales. So we have the molecular scale with the cytokine receptor interactions, TCR-MHC interactions, which Kathleen mentioned, the cellular scale, understanding the developmental trajectories, cell activation, differentiation, and then the whole organ scale that I described with this organ axis model where we're understanding the entire lobe of the thymus. And of course, then the whole body scale is relevant in terms of immunity and tolerance and all the functions of the immune system that I mentioned. So this is unpublished work, and I want to again say this has been a really international collaboration with Nadav Yayan, who was a postdoc in the group, and also shared with John Marioni's lab and Virginie Ullmann, and Veronica Kedlian, who was an amazing PhD student in the group. And this was a collaboration with Ron Germain at the NIH and Andrea Radke, who did the IBEX multiscale protein imaging, and the Notarangelo lab at NIH, and also Tom Tagan in Belgium. And of course, we must never forget the donors of these tissues, the women, the children, their families. These tissues come from children's hospitals and from the Human Developmental Biology Resource. And looking towards the future, you can see here that mapping the human body across scales, from sort of 0 dimensions where we started, through 2 dimensions in tissue sections, and then going up to large volumetric morphological reconstructions like with hierarchical phase contrast tomography, will give us new insights and valuable insights for a better understanding of ourselves, but also for drug development and drug discovery. And this multi-scale modeling and mapping is enabled by the Canadian Institute for Advanced Research and in collaboration with Gary Bader, who's a co-director of mine in that program, multi-scale mapping. And of course, he's based here in Toronto, and also Kati Bader in Indiana. These are my disclosures, and I will stop there, say thank you, and take questions.
Excellent. Thank you, Sarah. Really phenomenal talk. I just remind people to get your questions in via Slido. Maybe I'll start with where you ended, Sarah. Just Eric started out this morning talking about the ambitious journey of the Human Genome Project, and starting out with sort of a project that was hard to see through its conception, and then eventually sort of working through to the point where you were doing implementation. It strikes me the Human Cell Atlas is somewhat similar in terms of that scope and ambition and so on. You've presented a few cases here of really, really impressive work. We also mentioned that there's collaborators working in other spaces around the world in the consortium and so on. Can we just say a few words about kind of how the Human Cell Atlas is sort of coming together more broadly in terms of international collaboration, where it is on the journey, and maybe what some of the hurdles remain to kind of see it to fruition?
Absolutely. No, so thank you. I'd be delighted to do that. So the Human Cell Atlas is this grassroots international consortium that you mentioned, has over 3,000 members from over 100 countries around the globe, and everyone is welcome to join it. And there are a lot of members here in Canada that contributing. I've already mentioned Gary, but there are many others, Sonia McParland, who have been atlasing the liver. So this is really grassroots, and there's more information at humancellatlas.org/join-HCA, where everybody can join and register who's interested in this quest of mapping the human body at the molecular and cellular level. And the project is sort of organized into working groups and biological networks. Biological networks focus on the individual organs and systems in the body, and we've generated over 100 million suspension cell data points, and increasing spatial data is coming through. And we're basically in the phase of now assembling data objects for suspension cell data sets for individual tissues and organs. And we're releasing sort of gold standard data sets now on the Human Cell Atlas, data.humancellatlas.org website. So you can kind of think of that as like the golden path assembly of the chromosomes of the Human Genome Project, at least in a first draft. And that's focused on suspension, not spatial data. We're sort of developing these first drafts now. There's going to be a big publication bundle at Nature Publishing Group at the end of November. And we're then looking forward to kind of the next phase of the project, which will be a lot I hope, more comprehensive coverage of the body and much more high-resolution spatial genomics data coverage of all the tissues. So that's how I see it going, yeah.
Awesome. Yeah, really exciting initiative. I think we have time for one question here from the audience. And so this is asking you to comment on the implications of your work with placenta and immune tolerance and the potential for treatments of autoimmune diseases.
Potential for treatments of autoimmune diseases. I mean, I would say at the molecular and mechanistic level, a lot of the tolerogenic interactions like PD-1, PD-L1, and so on were, can be observed in the placenta. So I showed with the NK cells like ENTPD1, which is the enzyme that degrades ATP, it breaks down ATP, and so on. A lot of those, at the molecular level, there are tolerogenic kind of mechanisms that may be relevant in autoimmunity, but they're probably sort of deployed in a, they may be deployed in a different way in different cells and different sort of tissue niches in autoimmunity. But yeah, it's at the molecular level, there are a lot of mechanisms that are repeated again and again in different parts of the body in different contexts.
Excellent. Well, thank you very much.
Thanks.
Please join me in thanking Dr. Sarah Teichmann for her wonderful talk.